If I had to pick one feature of cells that was the most important difference between prokaryotic cells and eukaryotic cells, then it would be the organelles. Prokaryotic cells do not have membrane-bound organelles. No nucleus, no mitochondria, no Golgi apparatus, no endoplasmic reticulum. None of most of the organelles that we're going to talk about later in this video today. But probably one of the most staggering and obvious differences between these two cell types at first, when you first look at them under the microscope, is just size. In order to give you an appreciation for the size disparity between eukaryotic and prokaryotic organisms, I'm going to link you to a video by Journey to the Microcosmos, footage taken by microscopist James Weiss, that shows eukaryotic paramecia kind of cruising around a mass of purple bacteria. There are thousands upon thousands of these bacterial cells in this mass, and compared to the single-celled eukaryotic organisms that are really big by comparison. So check that video out, the link is in the description. So, eukaryotes, as I mentioned in the last video, eukaryote means true nucleus. True, karyot, seed or kernel, but it means the nucleus. It's referring to the nucleus, so true nucleus. Only eukaryotes have membrane-bound organelles, and on this diagram, these two diagrams, you can see lots of those different membrane-bound organelles. Prokaryotes have ribosomes, but ribosomes don't have membranes. Only eukaryotes have interior membrane compartments like these. These compartments are referred to as organelles, tiny organs. So just like you have a heart that does one job, and a liver that does another job, and a stomach that does another job, they have a mitochondria that does one job, and they have a Golgi apparatus that does another job, and a smooth endoplasmic reticulum that does another job. And you're going to learn what all these different jobs are. So first, let's just take a look at an animal cell. This is an idealized animal cell. At the bottom here, I've written "Ceci n'est pas une cellule," which means this is not a cell. This is a reference to a old famous piece of artwork called The Treachery of Images, which is a picture of a pipe, and it says this is not a pipe. The idea is don't confuse the image of something with the real thing. And that's the same mentality that I want you to bring to this image. There are no animal cells that look like this. There's no animal cells that has this giant cell body and this teeny tiny little flagellum there. And most animal cells don't even have a flagellum. Only one kind of cell in a human body has a flagellum, and that is a sperm cell. And then they wouldn't have most of these other materials inside them as well. This is an idealized animal cell. It is an illustration created in order to show you all of the different components that might be contained inside any given animal cell, but is not meant to represent any particular animal cell. This does not look like a muscle cell. This does not look like a bone cell. This does not look like a brain cell. This doesn't look like a skin cell. This doesn't really look like any of your cells, but it is definitively an animal cell. How do I know? There are no chloroplasts inside. Pretty good indication that it's an animal cell. Not definitive, though. A fungus wouldn't have any chloroplasts either. How else can I tell? There's no cell wall. That's a good indication that it is an animal cell. That's kind of unique to animal cells, actually, not having a cell wall. Plants have a cell wall. Fungi have a cell wall. Bacteria have a cell wall. Only animal cells definitively have no cell wall. So that's a pretty good indication right there. So, this is an idealized animal cell. What components does it have? Well, let's start with the ones that we saw in the bacteria cell already, the ones we're familiar with. It has a cell membrane, a plasma membrane, that oily outer membrane that all cells have here going around the outside, and that is the exterior portion. The purpose of the plasma membrane is to regulate what materials are allowed in and out of the cell in order to maintain the internal environment, which is part of the regulation requirement of life. It helps it maintain homeostasis. And one thing about cells is that they are able to accomplish all of those criteria of life that we discussed in Unit 1. So it needs to be able to maintain its homeostasis. Plasma membrane is the big way that it accomplishes that task. it has cytosol, right? All this lightish blue liquid that you see here, that's that honey-like or jelly-like substance of all cells. So you would find those in any kind of cell, that you would find that material in any kind of cell. Now, in eukaryotic cells, you call the compartment between the nucleus and the plasma membrane the cytoplasm. So that's essentially the cytosol and everything that's inside the cytosol as well. The cytoplasm is that compartment between those two sets of membranes. Another thing that we saw inside the bacteria cell would be ribosomes. And just like in the bacteria, you can see that there are ribosomes floating around in the cytosol here. But you also have ribosomes studded on the outside of this membrane here. And we'll talk about this membrane a little bit more in a second. That's going to be an important one. What else did we see inside the bacteria cell that you would find inside animal cells? Well, DNA, right? Now, in all eukaryotic cells, the DNA, or the bulk of the DNA at least, is going to be kept inside of the nucleus. And the nucleus is this large purple golf ball looking structure here in the center. And this kind of goopy, it almost looks like we've cut a jelly donut in cross section here. All of this purple goop in the center is the DNA. It's in the form of what we call chromatin right now. You can't see the individual chromosomes when it's in this form. It's kind of like spaghetti on a plate, right? If you know you have individual strands of spaghetti, but when it's all mixed up on the plate, you just see a big old mass of stuff on the plate. That's chromatin. Chromatin is a mix of DNA and protein. It's all just one big glob there that's contained inside that nucleus. So that's our container for DNA. And you can actually see that the nucleus has not only its own membrane, but it's actually got a double membrane. This double-walled membrane that we have here is what we call the nuclear envelope. So every membrane is a phospholipid bilayer. You remember that from the lipids video. But this actually has two phospholipid bilayers. This is a double membrane of bilayers. So the nuclear envelope keeps that nucleus safe. And that makes sure that you're not going to get like stray viruses coming in or stray enzymes like DNases that might come in and start eating away at stuff, because you want to keep the DNA nice and safe inside this compartment. Now, you might also notice that there are holes inside the nucleus because we do need to get the information out sometimes. We need to allow some stuff in and out. So the nucleus has what are called pores to allow information in and out. We'll talk about how that works a little bit later on. One thing that I think draws people's attention about the nucleus in this illustration in particular is this large, dense structure right in the center, which I don't have labeled here, so let me label it. It is called the Nucleolus. The Nucleolus. It's another structure inside of the nucleus, and it is made of DNA. It's made of chromatin, and it has a pretty high ratio of RNA in here as well. What the nucleolus is just a very, very dense region of chromatin inside the nucleus, and it contains all of the genes for making ribosomes. So all these little blue dot structures you see out here, well, you have to make those at some point. So all the information for making them, it's found inside this little nugget here, inside this cluster called the nucleolus, right here at the center. I'll talk a little bit more about that later, but we have these two structures here, the nucleus, which is the container for all of the DNA, and the nucleolus, which is part of the nucleus, which contains the genes for making ribosomes. One thing about biology that you just kind of have to learn to love is that sometimes you'll be handed 3 to 7 terms that sound uncannily similar to each other, so nucleus and nucleolus, chromatin and chromosomes, cytoplasm and cytosol. Sometimes you just have to memorize which one is which. And if you need help, come to office hours and we will work it out together. But in the meantime, let's work our way out from the nucleus, out from the nuclear envelope, and to this next network of membranes, which is a little bit purple in color and studded with ribosomes here. And I like that network of membranes. It kind of looks like a net, doesn't it? This kind of crisscrossing, intersecting pattern of membranes. What if we had a Latin-y word that meant net-like, like reticulum? Yeah, that's the Latin word for net, isn't it? And we could call it the net-like thing in the cytoplasm, the endoplasmic reticulum. Oh, you know what though? There's another net-like thing over here. There's another endoplasmic reticulum over on this side. If only we had a way to differentiate it. You know what? This one's got little rhinestones on it. It's got all those little ribosomes all over the place. It looks like if I ran my finger over it, it would be coarse in texture. Let's call this one the rough endoplasmic reticulum. And then the other one, we'll just call that the smooth endoplasmic reticulum. And that is more or less how these two structures got their names. They are the nets inside the cytoplasm, the endoplasmic reticula, and they are rough and smooth because they look rough and smooth underneath the microscope. Now, they have two very, very different jobs. The rough endoplasmic reticulum is directly outside the nucleus. You can see it's physically attached to the nuclear envelope. So any information that leaves the nucleus, any molecule that physically leaves the nucleus, is going to end up in the rough endoplasmic reticulum. And if it then bumps into one of the ribosomes that are here at the rough endoplasmic reticulum, it can then be translated into a protein. And the rough ER is made out of these liquidy membranes. And the wonderful thing about membranes is that you can pinch off part of the membrane in order to make a little transport box, a little package for you to ship elsewhere inside the cell. I'll go on more about that later as well. But it's a really neat little trick that cells do. They pinch off part of themselves and send it elsewhere. And each organelle can do this trick as well, at least some of the ones we're going to talk about right now. So the rough endoplasmic reticulum is a kind of labyrinth which catches any information that's trying to leave the nucleus and directs it over to the ribosomes, who have the job of making proteins, and then the proteins get shipped off by the rough ER to the next location for the proteins to get modified, which is the Golgi apparatus. Talk more about that in a second. Meanwhile, the smooth endoplasmic reticulum has nothing to do with protein synthesis whatsoever. No ribosomes involved. You can see it's completely smooth in texture. There are no ribosomes, okay? What the smooth endoplasmic reticulum does is it is a lipid factory. It makes phospholipids, it makes steroids, it makes all kinds of different lipids, so you can thank it for all of those nice lipids that you got going around in your system. It also helps to detoxify the cell. So any kind of circulating drugs that you have in your system, well, they have a biological half-life. They decrease in potency over time, which is why if you have a headache, you take some Tylenol, right? But eventually the headache comes back. You might need to take another one after a few hours. Well, part of the reason is because the smooth endoplasmic reticulum is detoxifying your system. Liver cells have a higher proportion of smooth endoplasmic reticulum for exactly this reason, because their job is partly to help detoxify your entire body, and they do that by utilizing the physiological capabilities of this particular organelle. And yet, somehow, this humble organelle seems to often go overlooked. So rough endoplasmic reticulum has ribosomes, ships polypeptides over to the Golgi apparatus for finishing touches, whereas the smooth endoplasmic reticulum performs lipid synthesis jobs as well as detoxification functions. Next organelle that I should discuss is probably this Golgi apparatus, since I already mentioned it, and it kind of looks like the other ones, so this will often trip up people when they're trying to do the labeling diagram. Notice that the Golgi apparatus is like the endoplasmic reticulum, but each one of these little membrane sacs that makes it up, these Golgi bodies, if you will, these cisternae, as they're called, they're separated from each other. They're actually, it's like a stack of pancakes, one on top of the other and top of the other, whereas for the endoplasmic reticulum, they're kind of interconnected. Also, the Golgi apparatus is entirely separated from all of the other structures here, right? The endoplasmic reticulum is connected to the nucleus, and both of the reticula are actually attached to each other, whereas the Golgi apparatus is over here. It's separated from the other two. So those are two good indicators that you're looking at the Golgi apparatus and not one of the endoplasmic reticulum. The Golgi apparatus has a receiving end and a shipping end. And what it does is it takes in polypeptides that were produced over at the rough endoplasmic reticulum, and it places them through its series of Golgi bodies, its series of cisternae here, and it puts on kind of finishing touches. It might add in a particular carbohydrate group where one might be necessary on the protein for some function. And then once the proteins are all finished, the Golgi apparatus's shipping side will then send it to its final destination, which let's say is just going to be embedded in the plasma membrane, the exterior membrane of the cell membrane there. So maybe it's going to be a transport protein that lets stuff in and out. In this way, you can think of the Golgi apparatus as being the post office of the cell, which is a pretty common way to think about it. Next, we have the mitochondrion, or mitochondria if we are speaking plural. And if you've ever taken any biology before, I'm sure when I say mitochondria in giant neon letters in your head lights up the powerhouse of the cell, which, yes, it is the powerhouse of the cell. Its job is to help release energy from carbohydrates and other types of high energy compounds. But simply knowing the phrase powerhouse of the cell is not going to be sufficient to pass any test that I give you in Biology 110. That phrase will not appear anywhere on any one of my tests, so you are actually going to need to know what a mitochondria does. So, for starters, its job is to manage the processes associated with aerobic cellular respiration inside of the cell, which is the process by which we take energy in larger macromolecules like carbohydrates or triglycerides or something like that, and we break it down into a much more ready-to-use form, which is a source of energy, chemical energy, that we refer to as ATP, adenosine triphosphate. Now, all of the nitty-gritty details of cellular respiration we are going to talk about, but it will be in Unit 3, so you don't need to know all of it just yet. What you should know about the mitochondria for this unit is that it handles the aerobic parts of cellular respiration for eukaryotic cells. You won't find these in prokaryotic cells. Now, if you take a look at this diagram, you see just a couple of threads in the background, these little gray threads here and there and all over the place. And you got these arrows pointing to it that says cytoskeleton. So just like we have a skeleton that holds us upright and gives us structure and allows us to kind of move around, like our muscles actually move us, but our skeleton, if it wasn't there, our muscles would have nothing to pull on, right? So our skeleton allows us to move. The cytoskeleton does the same for the cell. The prefix cyto means cell, so this is literally the cell's skeleton. And the cell's skeleton is made out of protein filaments, columns of protein. These are called microtubules and microfilaments, different kinds of protein threads running every which way inside of the cell. And they actually create this dense mesh, this dense network of proteins that hold everything together. Remember, the plasma membrane is an oily liquid membrane, and the cytosol has a viscous honey molasses jam kind of consistency. So we have a bag filled with jam that's made out of oil. Not a very structurally sound container overall. What's holding all of that together? Well, a lot of it is these cytoskeleton proteins that have enough structure and enough tensile strength to actually give this thing shape and to give it compressional strength and to give it tensional strength. Tensional strength means that it's hard to pull a cell apart, and compressional strength means it's hard to smash it, to squish it together. And those kind of things are important if, for example, you are lifting weights, right? And the tension of that weight is pulling on your muscle cells. You don't want your muscle cells to just tear apart on each other. It is these cytoskeleton fibers that are going to be holding all of your cells together. And these cytoskeleton fibers are going to be able to attach themselves between cells as well. Their parts of them are going to form connections at what are called desmosomes between the cells in order to strengthen the connections between cells in order to form tissues. Another function that the cytoskeleton serves is that it anchors all of the organelles in place, and the cytoskeleton can be used in order to transport materials as almost like a monorail system around the cell in order to transport material from, say, the rough endoplasmic reticulum over to the Golgi apparatus, because we have these lines of fibers going from one over to the other. So it's a transport network inside the cell as well. So a lot of cool things can occur because of the cytoskeleton. We'll talk more about them later. As always, we'll always have more to talk about later. Okay, so we got the nucleus, smooth endoplasmic reticulum, Golgi apparatus, rough endoplasmic reticulum, mitochondria, plasma membrane, cytoskeleton, ribosomes. Let's talk about the ones in this bracket here, things that you will not find in plant cells. So, what have we already learned? All of these organelles here, they will be found in plant cells. These three, though, these are animal cell exclusives. Number 1, the centrioles. Centrioles are tight little bundles of microtubule proteins, the same kind of proteins that make up that cytoskeleton. They don't do a whole lot right now, but they are very important when cell division starts coming up. So they will be crucial machinery for constructing the mitotic spindle, which is the structure that pulls chromosomes apart for cell division. So they will come up during mitosis. The lysosome, I love lysosomes, I think they're really cool. They are a little membrane-bound compartment that is filled up with digestive enzymes. They have lipases for lipids and DNases for DNA and amylases for starch, and they got all kinds of enzymes for breaking down molecules inside of them. So they're really, really good at eating away at materials. But you want to keep all of those enzymes contained. You want them inside that lysosome, because if you were to just release that into the cell, well, the cell's made of lipids, the cell's made of carbohydrates, the cell's made of proteins, and it's made of all of these other materials, so the cell would end up digesting itself from the inside out, which sometimes the cell might want to do that if it wants to self-destruct. Like, for example, if it was infected by a virus or it had turned cancerous, sometimes cells undergo what's called apoptosis, and they might want to do something like that, but most of the time they're going to want their lysosomes to stay sealed up and only use those enzymes when they want to digest something. But how do they use the enzymes if they're inside the sealed little compartment? Well, when food enters the cell, when it comes through this exterior membrane, it enters inside its own little pocket. When you pinch off a little pocket of the outside and bring it into the cell, it's called a vesicle or a vacuole. And you can merge that. You can push the lysosome and the vacuole together, and the interior contents will spill into each other without ever escaping into the outside of the cell, and they will mix together. So you'll get the digestive enzymes and the food mixing together. So the lysosome effectively becomes a free-roaming, floating stomach inside the cell, looking for food to digest. And once it finishes digesting whatever's inside that food vacuole, then the nutrients can diffuse out of that membrane, go into the cell, and then you can get rid of all of the waste by just kind of pushing it back out of the cell again. So the lysosomes are involved in digestion, and you find them in animal cells, not plant cells. Then you have this flagellum, which again, most animal cells don't have. Only the reproductive animal cells are going to have these. But importantly, no plant cells will have a flagellum. This is an animal cell specialty. Now, bacteria cells had a flagellum, if you remember, but they had a single protein filament flagellum, and they usually had more than one, and they spun around 360 degrees like a little propeller. Eukaryotic flagella, too big for that kind of thing. It's so big that if it tried to spin around like that, the shear forces would just pull it right off, and it would go flying and just be a big mess. It would probably tear the cell open. It's just too big of a structure to try to get that kind of mechanism going. So instead, it's using these microtubules, the same kind of proteins in the centrioles and the same kind of proteins in the cytoskeleton. It's using those microtubules in a particular configuration, like giant girders, and it kind of wags them back and forth to create a whip-like motion, back and forth and back and forth, and that whip-like motion propels, say, sperm cells forward so that they can navigate through the water. So flagella are an animal cell specialty, certain types of animal cells at least. Now that we've taken a tour of the animal cell, let's move over to the idealized plant cell. And I know, that was a big bunch on our plate to look at the animal cell, but there's good news. Because all eukaryotic cells are going to share certain characteristics in common, even more so than all cells in general share in common. So, a lot of the organelles that we just looked at are already here. We don't need to talk about them again. Plant cells have a cytoskeleton. Look, there it is. Plant cells have mitochondria. And make sure you take specific note of that, because a lot of times students get confused. They think, oh, plant cells have chloroplasts and animal cells have mitochondria. No, plant cells have both mitochondria and chloroplasts. Plant cells have a nucleus, and that nucleus has a nuclear envelope and a nucleolus inside of it. Plant cells have a rough endoplasmic reticulum. Plant cells have ribosomes. Plant cells have smooth endoplasmic reticulum. Plant cells have a Golgi apparatus, a plasma membrane, and a cell wall outside that plasma membrane. That's something we didn't have in the animal cell. And that cell wall has holes in it. Why do we have holes? Well, sometimes you need to be able to pass materials from one cell over to another cell, either for communication or to move nutrients from one place to another, or water from one place to another in order to maintain pressure from one location of a plant to another. Lots of reasons you might want that. So you need these little channels in between cells. And if you have a solid barrier in between them, that's hard. So you need these plasmodesmata, they're called, in order to get through the cell wall. So cell wall, that's something you'll see in plant cells, but not animal cells. The chloroplasts, that's something you'll see in plant cells, but not animal cells. Chloroplasts, of course, where you see photosynthesis taking place inside of plant cells. And then there's really only one organelle that we haven't mentioned whatsoever up to this point in the material, and that is this, the central vacuole. And it is a big ol' organelle, isn't it? Look at the size of this thing. It takes up more space than any organelle in the entire rest of the cytoplasm here. The central vacuole, well, first of all, a vacuole is just a container inside of the cell. It's just a membrane-bound compartment inside of the cytoplasm. You can have smaller vacuoles, you can have, and if they're really, really tiny, we refer to them as vesicles. But this big one right here is referred to as the central vacuole. And what it is mostly filled with is water. It is filled brimming with water. So much water that that water is under high pressure. So much water that it's actually pushing against all of these other organelles. It'll shove all of these other organelles against the rest of the cell wall, pushing them against the walls. And that is on purpose, because plants, think about herbaceous plants like dandelions and flowers and that sort of thing. Think about what happens when you forget to water them, right? They wilt. Why is that? Well, the only reason they're able to stand is because of hydrostatic pressure. They need water pressure in order to stand upright. And that water pressure, that water, is held inside this central vacuole, inside their cells. So they need to fill up this central vacuole with as much water as possible in order to stand rigid, in order to stand upright. So that is the main function of, not only as a reservoir of water, but also to allow them to stand upright and give the whole plant this kind of structure. Now, if the entire purpose of the central vacuole was just to fill it up with water and fill it up with water and fill it up with water and fill it up with water, and that's all there was, the cell would just kind of break open, right? It would be like trying to overfill a water balloon. The whole thing would make a mess. It would burst open. If we did this in an animal cell, that's what would happen. So there is an interplay of forces between the central vacuole and the cell wall. The central vacuole is pushing out with all of its might. It's trying to create as much pressure pushing outward as it can, whereas the cell wall is trying to keep everything in. And it is that opposition of forces, pressing out and pressing in simultaneously, that gives plants the ability to stand upright. That's where that turgor pressure comes from, their ability to kind of stand up against the force of gravity and hold their leaves up to the sky and show them to the sun and do photosynthesis at their maximum capacity. So the central vacuole plays a really big role. Now, interestingly, under a microscope, you really can't see the central vacuole because it's basically a big bag of water, and water is transparent. So if you shine a light through it, it just looks like a big old bunch of nothing. But if you know what you're looking for, chloroplasts have a nice deep green pigment to them, and the central vacuole pushes chloroplasts against the cell wall. So you don't look for the central vacuole. What you look for is where there is a void of chloroplasts, where there is an absence of pigment. That's your central vacuole. Okay, so that was my tour of the two kinds of eukaryotic cells that we're going to be labeling in diagrams. Sorry, we're not going to do fungi cells in this class. I wish we had time, but we're just going to do plant and animal cells. In the next few videos, we're going to talk about cell surfaces, cell membranes, the composition of cell membranes, and how materials get across those membranes. We're going to start talking about osmosis and diffusion and facilitated diffusion and active transport and passive transport and phagocytosis and exocytosis and all kinds of terms like that. And then after that, I will probably have some videos where I talk about some of these organelles more individually, or just a couple of them at a time, but I wanted to make sure I had at least one video where I gave you a big overview of, hey, here's what these cells look like, big picture view, and generally what each one of these organelles do. So I will see you next time.